CD. So have you connected this?
So I'm going to connect to the Zoom, right?
Yeah, connect. Yeah. So the microphone connect. Yeah.
And now everyone should be able to hear you.
Okay.
Hi, everyone.
Okay.
Yeah, please.
So we're 6 minutes behind schedule, but anyway, so the last speaker of this session is CD from University of Toronto. Okay, so you're gonna talk about statistical dynamics of tumor problem. Thank you. Yeah.
Well, thank you for inviting me. Thank you for the birthday boy. I don't know you, but you know, it's pleasure to always sort of celebrate these things, and you know, it's kind of fun to sort of look back. And thanks for inviting me. I'm locally here in Toronto. So I'll talk a little bit about some of you have heard this before, because I gave earlier, actually no one is here, which is common. Okay, which is good. Okay, then I feel less bad about talking about it. So we'll talk about some work on thinking about tumor initiation and how tumors respond to drugs. Right? So in a nutshell, what I want to just introduce what I'm thinking of tumor is, let's make some time axis and y-axis is, the idea is that we keep generating tumor cells all the time. Right? We're constantly generating tumor cells. Our immune system is eliminating that. Right? So that's what most tumors are. So if you sequence my skin right now, it's filled with tumor mutations. Right? People have seen this in many, many examples. Many often our tumors can exist in some weird equilibrium phase where they're kind of very slowly growing, rather than sort of the immune system is keeping in check, but it hasn't really quite exploded, right? Tons of tumors in that category. And the worst part is when the tumor really escapes, right? So it's sort of now the immune system can't keep it in check, and it's now blown up and is starting to sort of cause symptoms of various kinds. Most times the original tumors are actually pretty small, a centimeter or 2 centimeter. So in some ways it's kind of miraculous that it kind of has this massive symptom, but it kind of goes to different tissues, right? That's when it really becomes lethal. So you sort of have small things happening everywhere, and then, one or the other organ kind of gives out, right? So that's sort of how one can think about tumor. And this escape phase is precisely where huge amount of tumor heterogeneity is generated often. Right? Usually what happens is, there's some replication machinery gets kaput somewhere here, and that allows this thing to sort of really sample a lot of mutations, and something then takes off. Right? So there's some picture of a very heterogeneous tumor, and this heterogeneous tumor is sort of very interacting, and that's what I think some of you also work on, in the sense that, how does this sort of tumor cells talk to each other of different types now, because it's a heterogeneous population, and how does that really talk to the immune system, right? And there's a mechanical angle to it as well. In this interactions could be sort of things that are close to each other, far from each other, mechanics of the tissue it's arising, and so on and so forth. So what I want to talk about, the work which we did in last several years, was one is thinking about tumor heterogeneity in this part, and how that kind of informs the drug response of the tumor, right? And this was some work in collaboration with people here at UHN, and There's some other work which we did with people in at McMaster, which I don't think I'll have time to talk about, but I'll sort of briefly touch upon. And depending on how the time goes, given that I'm a little bit behind, I'll try to talk a little bit about this part where the tumor is initiating, where we usually have very little data. So here I'm going to focus on some theoretical model of thinking about how tumors initiate and how sort of microenvironmental feedback might play a role. Seems to be another important aspect of tumor dynamics. This is something we're still working on. I'll be happy to talk to people if they're interested on trying to really tease out that interaction map, but we'll see about that. Okay, let me start with the first. This was work done with Catherine O'Brien, who is a pathologist here. Sumi was a postdoc in Catherine's lab, and Sophie was a graduate student in my lab. Good. So what's the idea? So the idea is that you have a tumor which is very heterogeneous, represented by these sort of colorful cells. You give it drugs at this point in time, tumor regresses, or you give it drug enough that it regresses. So you keep upping different treatments, doses, and then of course we don't quite have a handle on what the regress rate look like, and this is what I'm going to end up talking about more. But you do stop treatment at some point because treatment is lethal for other stuff, right? It's toxic. And this is the part which we don't like about tumors when they relapse, right? And the hypothesis is that the relapsed tumor is sort of much less heterogeneous compared to what you started with, because there's some cells which are survivors, if you will, which can go through this process of treatment by drug. These are often called drug-tolerant persister cells, right, which are sort of sitting here. And the question for us was, what is the nature of them, and what is the diversity here? That's the kind of question we wanted to go after. So this was the experimental setup that Catherine and Sumi and others in her group did. They basically start, it's a xenograft model. They start with sort of some primary human colorectal tumor cell lines generated from patient-derived. So it's patient-derived xenograft model. Sorry, not a cell line, but it's sort of derived from the patient. You barcode them. So now you take each cell has an individual genetic barcode. So it gives it a color, so you can track its progeny. When you take those, put it in the back of the mouse, grow the mouse depending on the treatments and whatnot, and whenever you want to measure what the tumor looks like, you take the cells out of it, sort out the human cells from the mouse cells. I think there's a fluorophore in it as well with the barcode, so you can just sort out the fluorescent cells and figure out what's the clonal composition. And I'm going to use this word again, but the clonal composition I mean is how many cells of different colors there are, right? So what's the average dynamic? So this is showing tumor volume for different treatments. So for example, this is saline, so just salt water, so you're not treating anything, so it grows. DMSO is another control, and in this case, the dose of, for example, 5FU was not high enough that the tumor could still grow, so it didn't treat anything. These two treatments, CPT11 and FOLFIRI, is the one where the tumor starts growing. You treat it here, it regresses, and then you take the treatment off here, and this is the relapse. Good? So what do you expect? Well, you're giving a very heterogeneous tumor to start with, at least in terms of barcodes. These ones should be pretty heterogeneous, right? Not everything is going to take, but still you should have a good mix of what you put in. And what you expect is that these ones are the ones where you kind of have low diversity. And the hypothesis was we can look at them, figure out which barcodes survive, then go look in the cells with those barcodes, and maybe there's something special about them, and we can kind of find out what allows relapse to happen. And just to make sure to clarify, this is still susceptible to drug. So this is not resistance, this is relapse. So you can take these ones and again give them drug. If you give them drug, they don't grow, DMSO they grow. Right? So it's not resistance. There are no genetic mutations have arisen in this tumor to give them resistance to the drug. It's absolutely just, it's still sensitive to drugs, it's a relapse. Good, any questions? Okay, good. So what do? You go and sequence them. So this is going to be a little bit busy slide, so let me just take you through it. So here I'm on the top, I'm sort of giving you different treatment types. Here is the the number of cells corresponding to each barcode. Take the question. Yeah.
Yes, I have a question. Could you go back
Yeah.
to the distinction between resistance and relapse? I mean, there are cells of every color that are after the treatment, right?
So there's some
After-
color. We're going to find out how many there are, but yeah, there should be a mixture. Could be a
Okay,
mixture, could be just one.
okay.
Maybe there's only one of them, which is sort of very good at surviving treatment. But again, this is not resistance. In the sense, a resistant population would not be affected by drug anymore. Right? So we did some experiments where you keep the drug on. Right? You can keep the drug on forever. You don't take it off. Eventually, you see something growing. When you take those tumors and give them the same drug, it's almost like giving them saline.
Okay, thank you.
Good. That's a very important distinction. I think it's going to be useful. Yeah.
Sorry, I just have a question about
Yeah.
the tumor itself.
Yeah.
So, I know you mentioned that ideally the multi-clonal population would have high genetic diversity.
Right.
But did you guys do any experiments where you
yes,
like
yes.
check that?
Yes.
Yeah.
So this is genetically diverse.
Oh, I see.
Again, an important question. Yes. And you can see, so, but that I think you should ask the second question, well, compared to what? Right? So you can compare it to the resistant population. Right? So you can go and sequence the resistant population and compare the diversity of the resistant population to this. And you can see that once it goes through this, really the bottleneck of selection, the diversity wipes out. So of course, this is probably not as diverse as in the patient. So it's always, yeah, it's an interesting question of how diverse it is and compared to what. Yeah. Good. Okay. So again, this is basically each dot here is the number of cells of each barcode at time 0. Right? So basically each one is almost equal sized. You know, you see a little bit of a spread, but not too much. And now you go and sequence them, for example, under salt conditions, right? You just give them salt water as treatment, and you see, so these are different mice. Sometimes they might be same mice, different tumors for sure. You can see some clones become quite big, right? You see a diversity of sizes, and for salt water, you know, which clone become bigs should not be surprising that they're all different, right? Some chance going on which one gets big because salt is not really doing anything. So this is the base dynamics of the tumor growth, if you will. You go to DMSO, 5FU, OXA, these were the ones where there was no effect of the drug, if you will, right? The tumor just grew. You see sort of this huge variability in which clone gets big and how much and so forth. So our hypothesis was that in these treatments where you actually see a relapse, you should see a very consistent result of which clones get big. So what surprised us was that this half of the plot looks very much like the left half of the plot. So if I didn't draw the y-axis, you wouldn't be able to distinguish anything from anything, right? So this was the breaking of the hypothesis that there are some special cells which survive drugs. So how do we make progress? So first is the two things which I want you to take away from this, and I'm going to look at it again. You can see that the difference in small and large clones can be really huge, like 10 to the 5, right? And second is there's no real correlation from tumor to tumor for the dominant clone, right? So those two things sort of don't sit together usually. When you see clonal dominance, you imagine it's from selection. That would mean there's some correlation between who is dominating. At least between tumor to tumor, right? With the same treatment in the same initial condition. That doesn't seem to be the case. So what we did to just quantify this a little bit better is we can now plot, okay, I'm jumping a little bit, I'm plotting on the y-axis is the cumulative fraction of clones, and the x-axis is their size, right? So I take this data, I take one of the columns, right? And I calculate the size of these different clones, which are given by these circles, and ask how many clones of a certain size do I see? Does that make sense? Right? So I'm forgetting about the colors of them, I'm just caring about their size. And I put them on a distribution. So I find that, of course, here it's a cumulative, so I find how many clones are bigger than 10 to the minus 6. Well, all of them are bigger than 10 to the minus 6, so I start at 1, right? And as I make my size bigger and bigger, I see that, you know, by whatever, 90% or 95%, I've exhausted all the clones. So there's one clone, for example, for this blue line there's one clone of size, what is it? Yeah, almost 90%, right? Almost 95%. Good? Good to read this plot. So what was surprising is that not only, so this looks like a very crazy plot, I don't know what I'm going to see, but it looks like their distribution is actually identical, no matter what the treatment type is, no matter whether there's a relapse or not, right? So now we can hang our hat on something and try to model something, right? And you can sort of plot, you can kind of quantify these distributions. So this is the T0, this is what we put in, right? Some kind of gamma distribution. And it turns out that your the distributions that you observe are more like sort of have these power law tails, right? And this is pretty tight. It's sort of the variance here is, the slopes are between 0.3 and 0.5. And different PDX tumors gave different slopes, by the way. So the question for us was, how can you generate such large heterogeneity, where some clones are almost 95% as big, or these power law-like tails, while having sort of some kind of neutral dynamics, right? There's no selection operating in principle, right? At least it doesn't look like it. I'm skipping some slides in the middle for interest of time, but so what we found that a simple model where you allow growth to be not exponential, right? Which is not surprising. For example, on 2D plate, if something grows, it grows as a square root n, right? Because the growth is only at the boundary, right? Full volumetric growth is your exponential growth, right? So any interactions will allow the growth to be sub-exponential, right? And you can kind of quantify that in terms of this, this parameter alpha. If you take this simple model and you put in a bit of hierarchy in the sense that there is a stem-like cell which becomes a progenitor cell, and then it sort of grows out, right? So you put hierarchy and the sub-exponential growth, you can actually get these distributions with different slopes depending on what value of alpha you started with, right? So if it's an exponentially growing thing, you basically get this blue line, which is how T0 looked like. And as you put in different values, sorry, alpha 0, as you put in different values of alpha, you basically get this sort of slopes built into it. And one can do sort of simple theory to show that this kind of works out. So at least this gives you a sense of, that there's a chance that this is all sort of equipotent stochastic population rather than some selective dynamics leading to these clonal dominance. So you can see huge clonal dominance, but it's really coming from these interactions rather than selection, if you will. So that changed the question drastically, right? We were going to look for these special cells, but there are no special cells anymore, right? It looks like the diversity is almost as much as you started with, right? So there's no real difference between diversity here and here. Right? The distributions are identical, right? Which clone is on the top changes depending on the experiment, so I can't quite put my money on that. So then what sort of became was trying to really understand this state, because that's where seems to be where the magic is, right? How are the cells surviving? Is there something special about that state rather than a particular type of lineage or a particular cell, right? So it changes the problem for understanding the genetics or particular lineage or particular clones to a particular state of the system in which the cells are probably transitioning to. I'm calling it state, which is a little bit loose, so but hope it's not too confusing. And the literature calls this, as I said, drug-tolerant persister cells. So there's some surviving cells which are drug-tolerant and persister-like, that they can be around even when drug is there, right? And then they are the ones which are causing relapse. So what's the nature of these is what we wanted to inquire next. And so Sumi really set up a good amount of, so the earlier experiments were in mouse. Yeriko.
I was actually wondering, sorry, sorry to interrupt.
No,
so I was wondering, like, well, what is the order of magnitude for, what is the timescale for mutations in cancer cells? Like, what if you sampled them right after they survived their drug tolerance, are they like more or less, is there less heterogeneity? Are they like regenerating heterogeneity over time?
I mean, there's mutations going on all the time,
Yeah.
right? So imagine in this, once they've hit this spot, they've probably lost some mechanism of doing DNA replication. So they're generating new mutations a lot. So I think the point here is that first, they're not generating diversity in the direction which gives them the resistance. Second is it looks like it doesn't matter what treatment you do, that they kind of relapse in the same way. So the clonal diversity seems to be not caring about it. Absolutely. So it's good. It's a good question. It's not to say mutations are not happening. It's not to say the timescales are small. Mutations are happening. Each of the clone is probably becoming even more genetically diverse over time. But it seems like that maybe not the important liver, right? So if, for example, if there were some mutations that were important to allow them to relapse, then again diversity will go down, right? That clone will outbeat the other ones, but you don't seem to be seeing that, right? So it looks like the mutations are not the ones driving this phenomena, not that they are not happening. Good.
Yeah.
Good
Okay. I
question.
hope does this mean that the cancer patient after the treatment and eventually die of cancer?
Well, it depends what you do after relapse, right?
Yeah, that's what.
What you do here,
But you said that all red cells, right? All tumor cells, right?
They're tumor
So
cells, yeah.
No heterogeneity.
yeah, they have the same heterogeneity that you started with, more or less.
You go back to the.
Looks like you just go back to it, right? So yeah, exactly.
Okay.
So yeah, it seems like you're stuck in a cycle. So you want to really target this state if you want to do anything, rather than keep targeting this state, because this state doesn't seem to be changing. So imagine the heterogeneity had gone down. Then you can say, look, now I can target this because it's now a narrower target, right? So I'm going to go after it. That was the idea, but it didn't look like that. So then we started thinking about this one. Yeah.
Exist because we didn't have a drug that quenches the escape phase, that quenches the tumor size.
It does, right? So tumor size did go down.
Yeah,
If the tumor size didn't go down, absolutely, right?
but we still allow for the escape phase to be there.
Oh yeah, that has happened in the animal already, right? In the animal, in this case, was the human, right? So escape phase happened in the human, right? Now you took those things and you're just treating them. You're treating them outside in a mouse, but this treatment is happening outside. So you're absolutely right that all the things that drive this thing are there.
As long as the tumor size is controlled,
Yeah.
we are okay.
No, we are okay, but you have to take the drug out. That's the problem, right? The tumor goes down until you keep the drug, but it's also killing other stuff, right? That is really the problem. You're absolutely correct. If you could keep having drug while the tumor is down, you'll be happy. I mean, it'll come, the resistance will come, but that might actually take a long time, right? So we may be okay. We just need a factor of 2, right? If you get a tumor at like 50, you need a factor of 2. You don't have to fully solve it, right? You get it to 100, you're fine, right?
I may be mistaken, but
No.
if I think about the tumor
Yeah.
as a spring,
Yeah,
so if I exert pressure,
yeah,
now the tumor size goes to 0.
right.
But if I let go,
Yeah.
it comes back.
Correct.
So that's your relapse.
Exactly. But the question is, what comes back? Do all the cells come back?
Everything.
Yes. So that's the problematic, right? So we were hoping that not everything comes back. That was the hypothesis. Because everything comes back.
Turned out to be wrong.
Exactly.
Okay.
Absolutely. Absolutely. That's why this was interesting to do any kind of modeling after that, right? Because so good. So that's why we want to understand what is happening when we compress it. So if we can kill them when they are compressed, nothing comes back. Right? That's the next half of 5 minutes. Yes. Right. So then we wanted to look at this state which they get into once you give the drugs. So what Sumi did was basically did this experiment. Now this is in the dish, not in the mouse. You take your control. Is there a projector? No. Use this hook, right? So this is your control. Don't give any drug. This is when you do regrowth, which is the relapse, and this is again you have the resistant. Right? This here you can do in the dish, right? And you look at what the gene expression of these different types, different states are. Right? You want to see resistant cells, relapsed cell, DTP cells, and control cells. Good. That might tell us some idea of what's going on. So it looks like sort of all your regrowth and control and resistant. So these guys, they're all kind of clustered together. In the sense, it doesn't matter. Once the cells are growing, they all look the same. Doesn't matter whether they've seen the drugs before. Doesn't matter whether they've seen the drug and relapsed. Does not matter whether they've seen the drug long enough to become resistant. If they're growing, they look similar. right? The DTP state looks quite different. Good, so it has a very different gene expression. So now that gives you something to work with, right? And it turns out the DTP state has something which looked like these paused embryonic stem cells. So there's these old experiment where people did was basically can take embryonic stem cells and starve them. So they pause, they don't die, they pause, they wait until the food will come, and then they will start doing the process. And this is what also happens in these worms, C. elegans. This is called the Dower state. You can take a worm, you can starve it, it just waits. Once the food comes, it goes, right? So this Dower state and pause state are something which people knew, and it looked like sort of these DTP states have a high correlation with this pause state. So this is the embryonic signature, this is random signature, these are the different populations. So well, this is my DTP state, blue. These are my control, regrowth, and resistant. So all of them look similar. That was the promise. And your DTP state has sort of a high overlap with this embryonic pause state, which people understand a lot. Now you can do this beyond just one tumor. You can actually go and look at data for different tumor types. And you can see that what they call minimal residual disease, right? So people have been characterizing these cells once you have given the drug. So this is the minimal residual disease. This is the minimal amount that's left. You can look at the gene expression of those things, and they seem to also have a significantly high overlap compared to sort of parent tumor on this embryonic signature, right? So it looks like there is something consistent from tumor to tumor. It's not only just about colorectal, not just about this mouse model. It looks to be a little bit more general. So then this gives you actually, well, then this is what I think Sumi and Catherine did, is that, well, they even went and asked what happens with the patients. So they can actually take patients and segment them based on how much their cells show embryonic signature. And you can see sort of, you can show different survivals. And the numbers were actually pretty good. I mean, the number of individuals were actually quite high. I mean, in the end, of course, there are few, but you see sort of significant difference developing quite early on. Right? So it's early, you can figure out early. So then you can say, well, okay, it's doing this, but is there something within this signature that I can target? Right? So it turns out that autophagy is a big signature in this problem, right? So autophagy is almost as discriminative as the whole embryonic signal, if you will. So they thought that maybe we can target autophagy and figure out some way of doing it. So now that's a prediction, right? So the prediction is if I take my tumor cells, give them chemotherapy, they're going to go into this DTP state, right? If I can kill the DTP state by blocking autophagy, nothing should come back when I take both drugs off, right? That's the prediction you're making. So it's the order of drugs matter. If you give autophagy drug first, it doesn't do anything because they're not in the state where autophagy drugs could work, right? So this is the experiment that Sumi really did. Okay, again, it's a very busy plot. So here, okay, dashed line here is basically what happened when you only give chemotherapy and take it away. So you give chemotherapy, it regresses, you take it away, it grows. This is the autophagy drug, SBI, which basically doesn't do too much whether you give it or not. In both cases, it grows. And then this is the combination, the green lines, which you don't see, is that when you give both drugs together and you take them off here, you really see no tumor coming back, right? So either it stops them from getting into the DTP state, because autophagy pathways are important to get into it, or if they get into it, they can't sort of survive it because they need it to survive. So that mechanistic detail is not clear, but you can see this kind of two-drug module works, which is independent of the genotype, right? Which I think is interesting, right? And now, of course, Sumi is trying to make a company out of it, and because it sort of allows you to do something very non-tumor specific and things like that. So she has a startup she's trying to go after more targets, etc., etc. I feel like I'm running out of time, but coming back to this idea that this clone size. So again, I claim this clone size distribution you get because of interactions, right? So then we wanted to really test this idea if this is really true, and glioblastoma was a great example because apparently there are two types of glioblastomas: one local, one diffused. right? So the local one would have stronger interactions because they're all bunched together, and the global one should not, right? And the simple theory we developed, which I didn't quite show you in its detail, you actually get different answers for those distributions, and that's what actually looks like in the data. So you can actually see very different distributions depending just on whether the tumors are diffuse or local. So that tells you that there's, this interaction is an important component of understanding this clonal dynamics, at least. So that's the end of my first part. If you like, I can take a few minutes and talk about the second. If not, I'm very happy to stop here. Keep going? Okay, I'm happy to keep going. So this is the work we did with, I talked about. The second half was basically talking about this initial part, right? What is happening in this initial part, where apparently microenvironment feedback is crucial, right? So this is work done with Anton and Matt. He's another physicist in the group, and Matt was our graduate student. So this is completely theoretical, but the idea was in this part, there are two elements which have been talked about. One is there's epigenetic switching to precancerous state. Let me just take out this plot for a minute, right? So the idea is that it's not just driven by mutations. It's also driven by some cells switching epigenetically to this precancerous state. The second idea has been that there's local microenvironmental feedback which can sort of facilitate tumor progression. Right? There's a positive feedback. Once a few tumor cells comes in, they allow others to kind of roll into it. So we just wanted to add these two features. The older models were very sort of step by mutations. You get mutation 1, you get mutation 2, mutation 3, you get 3 mutations, you become tumorigenic, right? Here we are saying, well, either there's sort of more to it in this local chemistry which starts and which people have sort of studied. So what Matt liked to call is an XYZ model. So X is your healthy cell. Y is this precancerous cell which is switching back and forth epigenetically. There's no mutations. And then once you are a precancerous, there's some mutation which can take you to be a cancer cell. Right? And all of them have some growth rates. So what's the tumor feedback? The tumor feedback is that these rates of switching changes depending on how many tumor cells you have. So as you get more tumor cells, it enhances the rate at which X becomes Y and decreases the rate at which Y becomes X. Right? So it kind of stabilizes the precancerous state. And then they can keep mutating into Z. So it's a positive feedback. Right? So that was the simple model. Some bunch of equations, which basically, this is the growth rates, this is the switching back and forth, and the Y also, this is growth, this is switching back and forth. There's a bit of mutation which takes you into Z. And this term is just keeping the total population constant. This is basically just turnover. Good. So the feedback. So as a physical picture, this is what is happening. You have a healthy cell, precancerous cells, they're switching back and forth. Few precancerous cells get a mutation, become red, Z. Now this thing kind of changes the rate at which things switch back and forth and keeps stabilizing them. So more and more of these guys are arising. Just a simple picture, and wanted to understand how to think about tumor progression. So it actually has, so the simple XYZ model, one is basically, so if you take no feedback, then you actually have a phase diagram where it says, well, if the growth rate of C is pretty large, basically you get tumor cells in the end. Because once you have one, it just outcompetes everyone, right? But if the C is a little small, the other cells can keep surviving. Right? So the growth of tumor maps, not surprising. Okay? So this is how sort of this XYZ landscape looks like. This only has one steady state in this part. You have some mixture of X and Y, very little Z. And in this part, you basically go to Z. So everything becomes tumorigenic, and it's easy to understand when it happens. Once you put feedback in, feedback means now Z can control these rates, right? When you put feedback in, you actually get two more phases. The two phases are, in this phase, which was supposed to be all XY, no tumor, you actually start getting a bistable state. You can sometimes get a lot of tumor because it's a positive feedback. Right? Positive feedbacks give us bistability. Good, right? So that's what you see, not surprising. What was intriguing was there's another phase that opens up where this steady state kind of moves in the middle. And you get sort of an intermediate state where things can be stable, all X, Y, Z can be together. And this happens because in this part, right, this is opening up in this part, right? It's basically opening up where Z should have won. It looks like X, Y can also kind of come into the game. Okay, this is another way of looking at it. So what do we want to do? We wanted to find out now tumor initiation, right? How does tumor initiate? If you have this simple XYZ model, we say tumor initiates if Z gets another mutation, right? And the promise of positive feedback is you should always be faster than the base case, right? Because now you have a positive feedback. Remember, Z is kind of making it more likely. So if you look at this bistable situation, it actually turns out, so what I'm plotting here, sorry, I'm going to just plot this. So I'm plotting here is the mean first passage time. I don't know if that means something to people. It's basically saying, what's the first time that you see a cell with 2 mutations? Right? That's what we're calling a cancerous cell. Good. So the mean first passage time actually decreases a lot when you have feedback by at least an order of magnitude, depending on in smaller populations, right? So remember, tumor initiation is a game of small populations, right? It's probably happening in one of your crypts somewhere in your intestine, or some tiny, lung has a small alveoli, which is kind of got fibrosis for too long, and some mutations can accumulate. So at small cell numbers, you can see a huge difference. You can actually, so microenvironment feedback or this epigenetic feedback can actually enhance the rate of tumor generation by a factor of 10. Again, not that surprising. It's supposed to be positive feedback. What was surprising is, sorry, I'm going through the plots a little bit faster, is in this phase, you actually slow it down. Right? So remember what was happening in this phase. Just wanted to look at the phase diagram. So without feedback, it would run directly here. Because remember, without feedback, this part was in all Z. With feedback, this steady state got dragged down in the middle. Right? So now the Z state is kind of low from which the mutations have to arise. So you actually make it slower. And you can see that here all the transitions are when you reach the max Z. Here all the transitions are happening when you're stuck in this intermediate steady state. And that's the reason you slow it down. So microenvironment feedback is not necessarily going to make the tumor initiation, it's a positive feedback, yet it does not always make the tumor initiation faster. So that sort of was the surprising conclusion we had in this part. And we were looking for some data, and there's a lot of stories about this sort of neoplasia becoming, faster or slower in different tissues and things like that. We couldn't quite grapple with it because this early tumor data is quite lacking. But if people have ideas how to look for signatures of slowing down or speeding up and things like that, it could be fun to think about. And yeah, in some language of sort of population genetics, they have some stochastic tunneling, sequential fixation, and things like that. So we did a lot of theory on that. So if you're interested, So those were the two sort of parts which I talked about. This is sort of equipotent entry and exit. Looks like embryonic sort of diapause-like states are important to think about this. And here, basically, epigenetic feedback, can allow sort of can make us think about how to think about tumor progression. And positive feedback can sort of both enhance and slow. So depending on the ABC values, if you could do some intervention, you could actually slow it down while giving it a positive feedback, if you will. Okay, I think I should stop here. So yeah, and this part we're trying to work with some temporal data, and I can tell other people if they're interested. Okay. Thank everyone. Thank you.
Yeah.
Some old pictures.
Thank you for this wonderful talk. Yeah. So